Welcome to our keynote lecture of the Collaborative Research Center Origin and Function of Metaorganisms at Kiel University. It's 4 p.m. in the afternoon. It's 10 a.m. in New Jersey in the United States. It's a very pleasure and a great honor to welcome Professor Liping Zhao. He's our today's keynote speaker. And welcome, Liping. Thank you very much for joining us and sharing your research in these indeed confusing and very confused times and very special times. So it's the first keynote lecture which we are doing now online. I also want to say a very warm welcome to our audience, which sits either in their offices or sits at home. We have at the moment close to 80 listeners which are watching Professor Zhao's lecture. I would like now to introduce Professor Zhao and his background. He is chair of applied microbiology at Rutgers University in New Jersey. He's also the director of the Center of Nutrition, Microbiome and Health. Professor Zhao is at the same time distinguished professor of microbiology at Shanghai Jiao-Tong University and head of several clinical studies and supervises several clinical studies on microbiome in diabetes, in obesity, and in other physiological functions. Professor Zhao got his education in Jiangxi Agricultural University and got his bachelor in plant protection and his PhD in molecular plant pathology at Nanjing University. I mention that because you will see that all his research is focused on diet and plant components and the role and the connection to the microbiome. And I think the foundation to that goes back to his early days at Jiangxi and Nanjing. He then became professor at Jiangxi Academy of the Agricultural Sciences and soon after at Jiangxi University. And since 2001, he was professor of microbiology in Shanghai at the Shanghai Jiao-Tong University. Had several important administrative functions there as well, was dean and distinguished professor. And in 2017 was recruited to the United States and is since then, as mentioned, the professor for applied microbiology at Rutgers. Professor Zhao is a fellow of the American Academy of Microbiology. He's fellow of the Canadian Institute of Advanced Research. He is, in 2011, was elected the best teacher of the year in Shanghai. And he is also editor of ISMI journal. He is in the advisory board of the American Gastroenterology Association. And we are very honored to have him here today and to share his research. Last year on the Gut Microbiome for Health Summit in 2019 in Miami, he made a much quoted statement which says, diet is the major force in shaping the gut microbiota. And I think this is what he goes to tell us today. Liping, we are very much looking forward to your talk, and the CRC 11A2 floor is online but open to you now, and the title of his talk is Dietary Fibers and the Foundation Guild of Gut Microbiota for Human Metabolic Health. Liping, thank you very much for coming.
Thank you, Thomas. Thank you for your kind introduction and hosting the webinar. Very great opportunity to exchange our ideas and results on how diet may change the gut microbiota and affect our health, particularly metabolic health. So we know that the complexity and the density and the diversity of microbes in our gut is such that we can call it our microbial Amazon jungle. And so it's a very complex microbial ecosystem. And if you look at the gut, it's essentially a bioreactor. So all the bacteria in our gut, they need to grow all the time in order to maintain a certain population level in the gut, because our gut content is being constantly removed at a certain rate, like about 10% on a daily basis. So bacteria need to grow and to compensate that loss. And they need energy source and other nutrients to grow. And the nutrients are primarily from two sources. One is anything in our diet which is non-digestible or undigested would become nutrients available to some gut bacteria. And you also see mucin and select cells and other nutrient-rich compounds directly derived within the gut. And they also are sources which can support growth of gut bacteria. So the diet and nutrients derived from a gut combined together will affect who can grow and at which rate and to what high level. And so this is why diet can significantly change the composition of the gut microbiota. And when bacteria grow in our gut, they will release many potentially bioactive compounds. And those are either beneficial or detrimental. And so eventually those compounds could even get into our systemic circulation and directly impact the gene regulations and expressions of almost any cell in any organ in our body, even including the brain. And so it's no longer true that our genes plus the environmental condition we are exposed to will decide our phenotypes. It's actually our genome plus our microbiome and our environmental conditions, and that actually decide how healthy we are. So to maintain a certain population level, the most important nutrient bacteria require is the energy source. And the best energy source would be carbohydrates. However, carbohydrates which are digestible, like simple sugars and starch, they would be digested and absorbed by human and leave almost nothing available to gut bacteria. But there is a very special form and potentially large amount of carbohydrates which are called dietary fibers. They are non-digestible, and so they will escape the digestion and absorption by human host and then become available to gut bacteria. So many bacteria can degrade and ferment on dietary fibers as their energy source, while at the same time they release short-chain fatty acids such as acetic acid, butyric acid, propionic acid as side products to them. However, these short-chain fatty acids are even essential nutrients to human. For example, they provide energy to our colonocytes, and they can mitigate inflammation, and they can even regulate our satiety. So there are many functions which have been reported in the past several decades, how short-chain fatty acids play a very important role in many aspects of our metabolism, immunity, and physiology. So I would say dietary fibers and gut bacteria and metabolic health probably is the best example where how we have established a mutual relationship with gut bacteria. So I would like to focus on how diet fibers and gut bacteria and affect our metabolic health, particularly obesity and type 2 diabetes. And we know there is a devastating epidemic of obesity and also related metabolic diseases such as type 2 diabetes all over the world now. And not just in developed countries, but also in rapidly developing countries. And so there are many hypotheses about why so many people become obese and diabetic in such a short period of time. And is it because we have too much of something bad? For example, too much fat, too much simple sugars, too much easy carbohydrates? Or is it because we have too little of something essential, such as dietary fibers? So our hypothesis is that it is probably due to we have not enough dietary fibers. So if you look at studies on fossilized feces back in 10, 20,000 years ago of ancient human, and you can realize that our ancestors probably had 200 to 400 grams of dietary fiber intake on a daily basis. While the average modern human, particularly in developed countries and large cities, may average only 15 to 20 grams. So that's a huge difference. That means over eons, our gut microbiota had been co-evolving mostly with us in a context of a high-fiber dietary environment. But now, over the last several decades, or no more than 100 years, we just shifted to a very low-fiber dietary environment. And in China, there is a long tradition of using food as a medicine. And there is even an officially published list of plants by China Ministry of Health. It's called traditional Chinese medicinal foods. So the plants on this list can be used both as food by common people and also as medicine by Chinese doctors. And if you look at those plants, they are rich in polysaccharides, non-digestible but potentially fermentable carbohydrates. So you can also call them dietary fibers. And we also collected a fecal sample from all over China, 314 young, healthy Chinese, 18 to 35 years old, half men, half women, half rural, half urban lifestyle, and but with 7 different ethnic groups. And not surprisingly, the gut microbiota is mainly segregated based on ethnicity. And however, even though different ethnic groups has a distinct gut microbiota composition, and if you look at the potential functional bacteria, you realize that 9 genera of short-chain fatty acid producing bacteria made up almost half of the total sequence we generated. So this is an indication that among healthy people's gut, fiber utilizing short-chain fatty acid producing bacteria probably are the major members, or should be the major members of a healthy gut microbiota. There is also a very interesting systematic review paper published in Lancet last year, commissioned by WHO, on how dietary fibers, carbohydrates, affect human health. And they compound more than 135 million person-years of data from 185 prospective studies, and also data from 58 clinical trials with almost 5,000 adult participants. And to look at the impact of high fiber and low fiber diet on human health, the conclusion is actually very straightforward. And regardless of the major chronic diseases you look at, there is a 15 to 30% decrease in all-cause and cardiovascular related mortality, and also incidence of these chronic diseases like type 2 diabetes and colon cancer between the high fiber diet group and the low fiber diet group. So basically, there is across different chronic conditions, constant, stable health benefits of taking high dietary fiber and to human health. So why is that? And what kind of mechanism actually mediate such a widespread and stable health benefits of dietary fibers? Are there any potential that some short-chain fatty acid producing bacteria play a key role in mediating the health effects of dietary fibers? And yes, if you look at the studies on various chronic diseases, you realize that there is a pattern start to emerge that among many chronic disease patients, like type 2 diabetes patients and colon cancer patients, you see a constant decline or reduction of abundance and diversity of short-chain fatty acid producing bacteria, particularly butyric producing bacteria, and also, of course, the declined concentration of short-chain fatty acids in their gut. So this seems to be strongly correlated with chronic diseases. And so then the question is, how can we dissect out of such a complex system and identify the potential key members which may mediate the health effects of dietary fibers to human. So we need to probably emphasize several things. First, we need to realize that bacterial functions are strain specific. So that means if you look at members or strains in the same species, you realize that by definition, they are allowed to have up to 30% genomic difference. Bacteria in the same genus are allowed to have up to 70% genomic difference. Well, members of the microorganism species like human, we only have 0.1 genomic difference among individuals. So this means that bacterial species are much more genetically diverse and functionally heterogeneous compared with microorganism species. So we must go down to species or even strain level if you would like to identify the key players in the gut microbiota. We also need to realize that gut microbiota is a microbial ecosystem. And just like in macroecosystems, no members are independent from each other. And different members of a macroecosystem, they would work together to form a functional group called guild. Members in the same guild, they would work together and they thrive or decline together. So they show co-abundant behavior. And then the question is, are bacteria in our gut ecosystem also form a guild-like structure? And different bacteria may work together to form a different guild. And then different guilds may compete or cooperate with each other. So we need to identify the guild level structure of the gut microbiota. And we also know that from macroecology, not all the members are equally important to a macroecosystem. For example, in the closed forest, the tall tree species are the foundation species. So they play a very important role in structuring and stabilizing the whole ecosystem. They need to grow to a certain abundance level to close the system and stabilize the system. So if you lose the foundation species, you start to lose everything. And then the question is, in our gut ecosystem, are there any species or are there any members they work or guilds? They work as if they are the tall trees for gut ecosystem, gut forest, and play the foundation species role. Of course, these are the ecological and microbiological considerations, but the most important question we need to answer is the causality question. So you see difference between diseased patients and healthy patients. And diseased patients, they have declined, reduced abundance and diversity of short-chain fatty-producing bacteria. Is this because they are diseased, so they have a different gut microbiota? Or it is because they have a different gut microbiota, so they become diseased? Chicken or egg, which comes first? And we argue in this perspective article that we still need to follow Kocher's postulates, the great German doctor who established the germ theory of disease and established a set of rules for identifying the causative agents of a particular infectious disease. However, in the microbiome research, we probably needed to make two modifications. First, the relationship between gut microbiota and the human chronic diseases is not dependent on one single bacteria, most likely. So it's a polymicrobial situation. And second, it's a two-way street. It's not just some bacteria play a detrimental pathogenic role, and other bacteria may play a protective and beneficial role in chronic diseases. So you need to identify both bad bacteria and also good bacteria. So we need to do first the so-called microbiome-wide association studies so that we can identify all the members which are either positively or negatively associated with the disease. And then we need to isolate the associated members either into pure culture or into a consortium with defined membership. And then you can use those isolates, inoculate them into germ-free animals, germ-free mice, germ-free piglets, germ-free non-human primates, And then after you colonize the gut with defined bacteria from human, then you need to apply the environmental condition, which are known to induce chronic diseases. And then see if you can reproduce chronic disease with human gut bacteria in animal. If you can, you have a new type of disease model for human. And then you can elucidate the molecular crosstalk between the colonizing gut bacteria and the host, and eventually elucidate all the molecular chain of causation between gut bacteria and eventually the development of a disease endpoint. And after you finish all this, you can say that you have identified some gut bacteria which play a causative role in either aggravating, inducing disease or reducing and preventing disease. And so these bacteria and their effector molecules could become both targets for treatment and prevention, and of course as biomarkers for diagnosis and prediction. So the best strategy to identify causative members in a particular chronic disease from gut microbiota probably would be to do interventional studies. And so we actually use diet as a tool to induce changes of the gut microbiota, and then see if we can also change the disease in patients. And so before, during, and after intervention, we collect the urine, blood, and fecal sample, analyze all the molecular level variations in those three windows, and correlate them to each other and also to the clinical parameters. Hopefully this can help us identify all the relevant members, either detrimental or beneficial, and as the very important starting point for understanding microbiome's role in human health. So we designed a dietary scheme where we provide balanced macro and macronutrients to human, and but at the same time to include a high amount of very diverse, physical, chemically diverse dietary fibers. So we call it a Feed Me, Feed My Microbiome diet. So I myself was the first volunteer to try this diet, and I lost about 45 pounds in a little bit over a year, and then remained stable almost over 10 years now. And so I would like to share in the rest of my talk two particular papers, and one on childhood genetic obesity and one on adult type 2 diabetes, how we can use all the concepts and the methodologies that we discussed so far in understanding the role of gut microbiota in human diseases. So the first example is a clinical trial in patients' childhood genetic obesity called Prader-Willi syndrome. So children with this particular disease, they have a genetic defect in the number 15 chromosome from their father's side, and they were born with very low muscle tone. So they even couldn't suck enough milk before weaning. But after weaning, after they start to take solid food, they quickly develop a very severe form of hypophagia, and they feel hungry all the time, can never be satisfied with any amount of food, and so they quickly become morbidly obese. And this is the most difficult form of obesity to deal with. And surprisingly, when we did our hospitalized dietary intervention targeting the gut microbiota in Guangdong Women and Children's Hospital in Guangzhou back in 2010, and starting in 2010, and we were surprised to find out that 1/3 of the morbidly obese children in the program turned out to be PWS patients. But they responded quite well to the dietary intervention. And they also showed improved food behavior after we put them on the diet a few weeks. For example, this boy, 14 years old, 140 kilograms, BMI over 60, he was in the program for 285 days, and he reduced his body weight to 83.6 kilograms. And then he went home and continued the intervention at home. After 430 days, he came back for medical checkup. He was 73 kilograms. He lost almost half of his initial body weight and also recovered from almost all the metabolic problems. And then, so we recruited a group of 17 PWS patients and another group of 21 morbidly obese children without genetic problem, and put them in the program. Genetic obese children for 3 months and simple obesity children for 1 month. And then we collected fecal sample and urine blood fecal sample at the baseline and end of each month. So they showed very good response to the dietary intervention and improved their metabolic health and lost 10% body weight for 1 month and 20% initial body weight for 3 months. So then the question is, this is a self-controlled study. So it's not a randomized controlled study. And are there any evidence that this dietary intervention induced gut microbiota changes play a causative role in the improvement of the disease? So we transplanted the microbiota at the baseline and end of the trial 3 months after from the same individual to germ-free mice. And so we found out that all the red-colored data are from mice which received the baseline gut microbiota, while green-colored data are from mice which received after intervention microbiota from the same person. So basically, the pre-intervention microbiota induced first inflammation in liver, in gut, in ileum, and colon. And after 2 weeks, after inflammation was gone, they started to accumulate a lot of fat. And while the post-intervention microbiota from the same individual didn't show such effect. So this is an indication that even though diet can directly improve metabolic health, but the diet-induced metabolic changes may also at least partially contributed to the improvement of the disease. So then we did a metagenomic sequencing of all the fecal samples we collected. So those metagenomic sequence data set cover both inter-individual variations of the microbiome and naturally existing, but also intra-individual variations of the microbiome induced by dietary intervention. And so we produced about 80 million short reads using the Illumina HiSeq platform, and about 8 to 10 gigabase pairs data. After we assemble everything into contig, we recognized about a little bit over 2 million non-redundant microbial genes. So this is a 2 million variables data set, but we only have 109 samples. And you must reduce the dimensionality and the sparsity of the data set if you would like to identify meaningful patterns. And so what we do is based on the genetic principle, we know that genes located in the same genome would behave in their abundance exactly in the same way because of genetic principle. So we do co-abundant analysis at the individual gene level. So we identified a little bit over 20,000 co-abundant gene groups. Each such group corresponds to potentially a genome or part of a genome. And then among these co-abundant gene groups, and 376 contain more than 700 genes, and they are potentially bacterial genomes. And then among these bacterial genomes, 161 show up in more than 20% of the samples. So, and they also make up about 60% of the total sequence we generate. So that means these 161 bacterial genomes already represent both the predominant members and the prevalent members of the gut microbiota among our patients. And we can even assemble high-quality drafted genome from each such CAG. Almost 80% of the genomes, CAGs, can be assembled into high-quality drafted genome. Okay, so now you reduce the dimensionality of the original data set from 2 million to 161. We have 161 individual genomes representing 161 prevalent and predominant gut bacteria. And now we can reduce the dimensionality based on ecological principle. So if gut bacteria form guilds, some of the bacteria will stick together and show their co-abundant behavior, and others will stick together to form another group. So we did a co-abundant analysis among the individual genomes, and we deleted them into 18 what we call genome interaction groups. And each such group is potentially a guild in ecology. So the same colored genome, they show highly correlated behavior in their abundance, and they increase or decrease together, so they form a guild. Then different guilds, if they are connected with red line, they also co-occur. But if they are connected with the blue line, they co-exclude from each other. After we organize the individual prevalent and predominant members into guilds, and now we can actually lump all the guilds in the same, lump all the genomes in the same guild to get a guild level abundance. And now we have 18 variables now. And when we correlate the guild level abundance with all the clinical parameters, 3 showed negative correlation with many clinical parameters, and 9 showed a positive correlation with clinical parameters, and the rest 6 showed no correlation. So now you have a network like this. Not only we know who works with whom to form a guild, but we also know each guild's relationship potentially with the host. The blue-arrowed guilds, they were reduced by the diet, they were promoted by the diet, and they showed a negative correlation with disease parameters. So they are potentially beneficial to the host. While the red-arrowed guilds, they are reduced by the new diet, and they showed a positive correlation with disease, so they are potentially detrimental or pathogenic. While the rest guilds, they showed no correlation with disease, so they are potentially commensal, neutral to host health. And so now we have all these up till now are just association studies, but they are association studies by reducing dimensionality of the huge data set based on genetic principle and ecological principle. And so this is an indication that major members of the gut microbiota may work together to form a functional group called guild. And so they behave like an ecological guild. And if you look at the current taxon-based analysis, you would realize that members from the same taxon, because even in the same species, they have a highly diverse genetics and potentially highly diverse functions, so they probably could not behave in the same way. And for example, if you look at the taxonomic composition of our guilds, you realize that some guilds, they even have members from 4 different phyla. And that means the guild is not taxonomically homogeneous. And bacteria organize themselves into guilds, into functional groups, not respecting our taxonomy. So we cannot use taxonomy and to understand gut microbiota, because each taxon is not, most often, is not functionally homogeneous. Even in the same species, for example, and if you look at the 5 genomes in the butyric-producing Eubacterium elegans species, they actually belong to 3 different guilds. And one guild negatively responding to the diet, and one guild positively responding, the other guild not responding. But if you combine them together to look at the species level changes, it's like this. It's not reflecting any real changes in the data set. So basically, we need to emphasize that gut bacteria probably function as guilds, not function as genus, as a family, or any taxa. And so we need to focus on looking at the ecological behavior of gut bacteria and their guild structure in order to understand who might be important in the disease. So the second example is a clinical trial conducted in adult type 2 diabetes patients. And we recruited a group of type 2 diabetes patients and randomized them into a control group and the high fiber diet group. And the control group is the so-called EuroCare group. Their diet is managed based on the advice, clinical guidelines from China Diabetes Association, which is pretty much copied the American Diabetes Association guidelines. And but we did a careful design so that the macronutrients, the calorie intake and the macronutrients intake between these two groups are essentially the same. But the only difference is the treatment group has nearly 40 grams of extra diverse fiber intake than the EuroCare control group. And this clinical trial conducted for 3 months, and we did a medical checkup, collected urine, blood, and fecal sample at baseline and end of each month. So basically, if you look at the primary outcome, and the treatment group had extra clinical benefits. So that means because of randomized design, if there's any extra clinical benefit in the treatment group compared with the control group, you can only attribute that extra benefit to the extra intake of the dietary fiber. So this actually confirmed many other clinical studies showing the clinical benefits of dietary fiber intake. So it's not surprising, but it's a further confirmation of the causative role of dietary fiber intake in improving human type 2 diabetes. We know that dietary fibers cannot be absorbed, digested and absorbed by human. So it's very hard to understand the molecular mechanism why dietary fiber can improve type 2 diabetes. And however, dietary fibers are the best energy source for many gut bacteria. So they may actually promote the growth of such bacteria. And so they induce the dietary fibers can induce changes of the gut microbiota. So would the change be causatively contributing to the disease improvement? So we transplanted again baseline gut microbiota and 3 months after intervention microbiota from the same individual to germ-free mice. And not surprisingly, the baseline type 2 diabetes gut microbiota actually increased fasting glucose level and disrupted the glucose homeostasis in the recipient mice, while the 3 months after intervention microbiota from the same individual didn't do such harm, actually improve the glucose homeostasis. So this is an indication that dietary fiber induced changes of the gut microbiota. First, the baseline gut microbiota may be aggravating chronic type 2 diabetes, while the 3 months intervention after intervention microbiota and may be protecting or benefiting the type 2 diabetes patients. So then we did a metagenomic sequencing of all the 172 clinical samples. Again, because of the clinical design and interventional design, such a data set can capture molecular level variations of microbiome, both across different individuals that's naturally existing, and also within the same individual that's induced by dietary change. And so we eventually generated a microbial gene catalog of nearly 5 million and 172 samples. So this is even bigger data set, even more variables, and very difficult to analyze. And however, if we look at all the genetic variations of the microbiome, using that to look at the structural variations, global structural variations of the gut microbiota during the intervention, you can see that at the beginning, there's no difference between the two groups. But after 28 days of intervention, they both different from their baseline and also become different from each other. However, after another 2 months, there's no further change. So this is an indication that high fiber diet can induce changes of the gut microbiota, and it takes about 1 month to stabilize the change, but there is no further change after that. And we also look at all the clinical parameter changes and also the global variations of the gut microbiota. There's a correlation between these two. So that is an indication that the more you change the gut microbiota, the better you improve the disease. And now we need to try to understand the mechanism. So basically, dietary fibers themselves may have some physical effect, like improve the regularity and however of the bowel movement. However, it's very difficult to explain everything just based on the improved regularity. And so the potential for gut microbiota changes induced by dietary fibers may be through short-chain fatty acids produced by fermentation of dietary fibers. So we look at the changes of 6 short-chain fatty acids during the clinical trial, and 4 of them actually reduced in both groups. So this indicates that they could not be responsible for the improved clinical parameters. Well, if you look at acetic acid, they showed no difference between the 2 groups, and also it showed a slight increase and maintained original level throughout the trial. And if you look at the butyric acid, there is no change in the control group, but there is a significant increase in the treatment high fiber group. So this is an indication that maybe the commonly shared clinical benefits between the 2 groups may be mediated by acetic acid, while the treatment group specific actual clinical benefit may be mediated by butyric acid. And we know that from many studies, acetic acid, particularly butyric acid, can bind to GPCR41-43 receptors from the L cell in the gut and stimulate production of a gut peptide hormone called GLP-1. And GLP-1 can actually stimulate insulin secretion. And so we see increased GLP-1 production in the treatment group and also increase insulin production. So this might be one mechanism that dietary fiber, after fermented into acetic and butyric acid, can increase GLP-1, increase insulin, and then benefit type 2 diabetes. If we stop here, there's nothing surprising, nothing actually new. However, if you look at the gut pH, you realize there is a significant reduction of gut pH. And so we know I'm a microbial ecologist. So when I look at the gut pH reduction, I realize there must be a profound changes of the gut microbiota environment. And so we need to look at the changes of all the major members of the gut microbiota. And again, we did a first co-abundant analysis among individual genes, and then group them into co-abundant gene groups. And then we identified 422 bacterial CAGs, each containing more than 700 genes. And 180 of these CAGs shared by 20% of the samples. So they are potentially both prevalent and also predominant. And then we try to do assembly of high-quality draft genomes. And eventually, out of these 180 prevalent and predominant members of the bacteria, and we assembled 154 high-quality draft genomes. If you look at these high-quality draft genomes, they already had more than 95% of their genes. And among these, 141 genomes actually harbor at least one of the key genes for short-chain fatty acid production. So genetically, these 141 genomes should be able to ferment some kind of carbohydrates and produce short-chain fatty acids. And they have this genetic capacity. But surprisingly, when we provide a high amount of very diverse dietary fibers to the gut microbiota, there's only eventually 15 such genomes actually promoted and become even more predominant, and 79 showed no response to the externally provided energy source. Well, 47 actually reduced even lower than the baseline. They become negative responders. So this is, and there are positive responders and negative responders, and they showed a negative correlation between them. And so these 15 positive responders, they all can produce acetic acid, but 5 of them can also produce butyric acid. While members of this negative responder group, and many can produce indole, which is a toxic compound, and also hydrogen sulfide, and many are gram-negative opportunistic pathogens, so they can produce endotoxin. When we look at the genome, we realize that there are many features which probably decide why some become negative responders and others become positive responders. For example, in the positive responders, they have much more proportion of genes devoted for utilizing starch, inulin, and arabinoxylan, which are plant carbohydrates. And they can also produce more energy out of the same amount of carbohydrates. And particularly important is they are tolerant to low pH. So they can continue to grow when they have acidified the gut environment by producing short-chain fatty acid. While the negative responders, they have higher proportion of genes devoted for using mucin carbohydrates, for example. So they are better at utilizing animal carbohydrates from the gut, and they are not tolerant to low pH. They would stop growing when the pH is lower than 6. And so all these combined together may explain why these 15 become positive responders and these 47 become negative responders. And we also show that if we combine the abundance and the diversity of the 15 positive responders and make it into an active short-chain fatty acid producer index, and we can say that this index is actually negatively correlated with the hemoglobin A1C. And that means during the first months of dietary intervention, the higher the abundance and the diversity of such 15 bacteria positive responders you can achieve, the lower the HbA1c after another 2 months for the patient. We also isolated bacteria corresponding to the genomes in these 15 positive responders. For example, this strain isolated and can actually reduce high-fat diet-induced obesity in mice. And it can also reduce fasting glucose level in germ-free mice. So germ-free mice taking the gut microbiota from type 2 diabetes patients showed elevated fasting glucose level. But if you combine this diseased gut microbiota with this particular strain, and it can significantly reduce the fasting glucose level. So this is another piece of evidence to show that these 15 positive responders, at least some of them, are indeed causatively contributing to the reduction of the disease. So basically the hypothesis is that the 15 positive responders, and they actually can produce acetic acid and butyric acid, and they acidify the gut environment, producing antimicrobials, occupying available niches. All the ecological effects combined together lead to decline of negative responders, and many of the negative responders can actually produce indole hydrogen sulfide, which can inhibit L cell from producing GLP-1. Endotoxin producers, which can induce inflammation, damage the insulin receptor, so increase insulin resistance. So when you decrease this, you release more GLP-1, you actually increase insulin receptor sensitivity. So all these combined together, direct effects on increasing GLP-1 from acetic acid and butyric acid, and the ecological effects leading to decline of the potentially detrimental and pathogenic guild, and may combine together explain why you see dietary fibers can elevate type 2 diabetes. So we hypothesize that foundation guild, the 15 such positive responders to dietary fiber intervention maybe play a role of the foundation species. Because it's not a single species, it's actually a guild, so we call it a foundation guild. And it's important for structuring a healthier gut ecosystem. Obviously, different people have different members to be their foundation guild, and some even lost most, if not all, the members of their original foundation guild. So you need actually design different combinations of dietary fibers to support and recover and maintain the foundation guild, because the same dietary fiber fermentable by my foundation guild may not be fermentable by your foundation guild. So this is where personalized nutrition may come into play. In summary, the foundation guild of the gut microbiota may mediate the benefits of dietary fibers to human health. Gut bacteria do not function as taxa, but as functional groups called guilds. Foundation guild is a guild that contains the foundation species of the gut ecosystem, which can structure and stabilize the gut microbiota. So the foundation guild may produce bioactive compounds, for example, short-chain fatty acids, that can both directly benefit the human host and also ecologically change the environment and keep pathogen guilds at bay. If you can design a high-fiber diet or high-fiber formulation which can selectively promote and sustain the foundation guild for each person, you may help people achieve their health on a personalized level. So I would like to acknowledge the contributions of my students, my colleagues, and our collaborators, and also the funding agency. Thank you, Thomas. Thank you, everybody. Thank you.
I would really like to thank Liping for a wonderful, interesting, stimulating, thought-provoking talk. It's usually now that we are going out for dinner and continue our conversations. This is not possible these days. Thank you very much for that wonderful talk. Thank you.